DNA bases are separated by only 3.4 angstroms, the width of a few atoms. DNA sequencing methods cannot easily resolve this dimension. Our sequencing by expansion technology, or SBX for short, overcomes this limitation by creating a surrogate molecule called an expandomer. This molecule is more than 50 times longer than its target DNA and encodes the DNA sequence information in large, high signal-to-noise reporters. The building blocks for the expandomer are novel Stratos Genomics probes called X probes. Expandomers are formed in a modified DNA replication process where X probes sequentially link along a target DNA template using a proprietary enzymatic process. This forms a double-stranded helix. Each X probe in the library has a unique combination of the 4 DNA bases depicted as A, T, C, and G. A tether is looped between the second and third bases and supports 4 high signal-to-noise reporters that mirror the X probe's base sequence information. Only an X probe with 4 bases that correctly matches the DNA template will be incorporated. The sequence information of the DNA is now expressed in the reporter sequence of the X probe. This cycle of linking X probes continues sequentially and extends along the entire length of the DNA. After assembly, the X probe linkage is released from the target DNA by denaturing, and a reagent is added which breaks a cleavable bond between the second and third bases of each X probe, allowing the backbone to expand. This product is called the expandomer. The expandomer sample is now enriched for high-value, long read lengths. Stratos Genomics will capitalize on a decade of nanopore development that has tried unsuccessfully to directly sequence native DNA. By scaling up size and signal of expandomer reporters, SBX technology enables nanopore-based DNA sequencing. In a nanopore detector, an expandomer chain is threaded electrically through a silicon chip nanopore. As each reporter passes sequentially through the pore, the electric current is impeded, providing one of 4 signals that uniquely identifies the next base in the DNA sequence. Each single silicon chip can contain thousands of nanopores, leading to measurement rates in excess of 10 million bases per second. Stratos Genomics brings you the basis of life.